Glucagon is a hormone produced by the alpha cells of the islets of Langerhans, as we found in the previous slide. The function of the glucagon is to increase the blood glucose concentration, especially in between the meals. And in this respect, it is just opposite to that of insulin. And of course, this also is a polypeptide hormone. And upon injection of glucagon into an animal, suddenly the concentration of glucose in the blood increases. So this is a hyperglycemic hormone. Glucagon is a hyperglycemic hormone. Mainly, glucagon is exerting its effects in two aspects. One is by means of breakdown of glycogen, that means glycogenolysis. The second one is increase the rate of gluconeogenesis. And you know that both these processes ultimately result in increased glucose concentration in the blood. And how is glucagon promoting glycogenolysis? First of all, glucagon is binding with its receptor on the plasma membrane of the liver cells, and this receptor is coupled with the adenylyl cyclase in the cytoplasm or on the cytoplasmic side. This adenylyl cyclase is catalyzed in the formation of cyclic AMP from AMP, and this cyclic AMP is activating a protein called a kinase regulator protein, and which in turn is activating protein kinase. This protein kinase is activating phosphorylase B kinase, and this phosphorylase B kinase is converting phosphorylase B into phosphorylase A, and this promotes degradation of glycogen into glucose 1-phosphate. And we know that from this glucose 1-phosphate, by means of the phosphatase enzyme, the phosphate group is removed and glucose is released into the circulation from the liver cells, and this is increasing the blood glucose concentration. Actually, this is a cascade system, which means at every stage the intensity of the reaction is or magnitude of the reaction is increased several times, and this means that each of the product is obtained or produced in a much larger quantity than the previous product. So this is a type of amplification mechanism. This is how only a few milligrams of glucagon can cause the release of hundreds of thousands of milligrams of glucose into the blood of an animal. And what about gluconeogenesis? You know that gluconeogenesis is the formation of glucose from non-carbohydrate sources, and glucagon is inducing gluconeogenesis when all the glycogen in the liver has been exhausted under the influence of glucagon itself. And this is going to happen especially if food is not taken in and if dietary carbohydrates are not obtained. And then you see most other effects of glucagon occur only when its concentration is rising above a maximum normal found in the blood. And one of the most important effects is that the glucagon activates adipose cell lipase enzyme, making increased quantities of fatty acids available to the energy systems. You know, if fatty acids are available for the generation of energy, then for those tissues where it can be utilized, carbohydrates can be spared so that The carbohydrates are exclusively available for those tissues which can use only them for obtaining their energy. Glucagon also inhibits the storage of triglyceride because this hormone wants triglycerides to be mobilized for the generation of energy. Glucagon also enhances the strength of the heart. We mean heartbeat. Increases the blood flow and especially in kidneys. Enhances bile secretion and inhibits gastric acid secretion. So they are of only minimal importance in the normal functioning. And once we get increased blood glucose concentration, for example, after a meal, this increased blood glucose concentration inhibits glucagon secretion. The effect of blood glucose concentration on glucagon secretion is just opposite to that of insulin. We know that insulin and the glucagon are antagonistic in function, which means they are functioning in opposite directions. And 90 milligram of glucose per 100 ml of blood is actually the minimum permitted level of glucose concentration in the blood. Once the glucose concentration is falling down to that much values, it can trigger the production of glucagon hormone. The opposite also is true. Once the glucose concentration is rising above 120 milligram per 100 ml, naturally it may shut down glucagon producing tendency. And again, increased levels of amino acids in the blood stimulate glucagon secretion. High concentration of amino acids must occur in the blood after a meal, stimulate the secretion of glucagon. And there are at least some instances where glucagon and insulin are not opposite to each other, and this is one of such an example. Because insulin actually increases the availability of amino acids etc. in the cells, uptake of glucose in the cells. And you see this influence of glucagon is at par with that of insulin. They are not opposite to each other. This is an exceptional case actually. Exercise stimulates the secretion of glucagon. A beneficial effect is that this glucagon prevents falling down of blood glucose level, so that during exercise carbohydrate is available as an immediate source of energy. This is a graph. On one axis we have the glucagon concentration, on the other axis we have the blood glucose level. You see, as the glucagon concentration is falling down, here this much is the glucagon concentration. Accordingly, you see here what happens. Try to explain it yourself. How many times glucagon concentration is present in the body compared with the normal. This is a good graph actually. Summary of blood glucose regulation. Both insulin and glucagon together are responsible for maintenance of the glucose in the blood. Whenever extra glucose is present, it is converted into glycogen, lipids, proteins, etc. under the influence of insulin. Whenever the amount of glucose available in the blood is less than normal, glucagon is secreted. This hormone is responsible for the mobilization of glucose from glycogen, from lipids, from proteins, etc. Not that much about these two hormones.